Biology Part 205. Each nucleus is formed around a specialized chromosome region called a nuclear organizer. Interphase is divided into three shorter stages, respectively G1 gap1, S synthesis, and G2 gap2. The duration of G1 is very variable and determines the time taken for cell multiplication of different tissues. Near the end of G1, a proportion of the body cells passes a critical point called the restriction point. When this happens, the cell becomes committed and will complete their cycles and divide to form new cells regardless of external conditions. Any cell which does not pass the restriction point is unable to divide. In most organisms, differentiated tissue cells lose the ability to divide and remain locked in G1. As these cells die, they are replaced by division from undifferentiated cells called stem cells. The duration of S and G2 are far less variable. Two important events occur during the S or synthesis stage. First of all, cell's DNA undergoes the application by the semi-conservative process described in detail. When the replication is complete, each chromosome consists of two identical molecules of DNA being side by side. This copying process provides the two complete sets of the genetic instructions, which will eventually pass into the daughter cells. The second event is the duplication of the centrioles. These structures will later migrate towards the opposite poles of the cells and may develop in the formation of spindle, although their precise function is not understood. The stage called G2 represents the cell's final preparation for division and involves a synthesis of histone and spindle proteins. Interphase normally accounts for at least 90% of the total cell cycle. The remaining events are concentrated into comparatively short divisions or M phase, typically lasting 1 to 2 hours. This short period may be further subdivided into 6 stages, namely the prophase, prometaphase, metaphase, anaphase, and telophase, and cytokinesis. So I can give them again: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Strictly speaking, mitosis includes only the first 5 of these stages, corresponding to the process of nuclear division. The 6th stage, called the cytokinesis, refers to the cytoplasmic division of the separation of the daughter cells. It is important to take note that this distinction, for there are many, is not directly followed by the cytoplasmic divisions as in many fungi. So it could have been the case there. The events taking place in each of these 6 stages are summarized here. So I'll give you the details. Prophase, the transition of G2 to prophase is not a sharply defined event. The chromosomes slowly condense, that is, they shorten and thicken, finally become visible under the light microscope. Each chromosome can then be seen to consist of sister chromatides linked together by a structure known as centromere. The two chromatids correspond to two identical molecules of DNA produced earlier during the S stage. Each chromatid contains all molecules of DNA and each is an exact copy of the other.